A very good day to my lecturers and my fellow friends. I'm Dharmin Ram Jagadisan with the matrix ID 193671. He is going to present a research article titled Isolation, Characterization, and In Vivo Efficacy of Escherichia phage myPSH1131. I extracted this research article from this PLOS ONE. Before going to the research article, let's peep a little on the backgrounds. Bacteriophages, commonly known as phages, which are viruses that infect and replicate only in bacterial cells. They have a wide range of applications that can be used in food processing industry, water treatment plants, against plant pathogens, a disinfectant for diagnostic tools, and a therapy to treat bacterial infections because they are known as a potent antibacterial agent. Why so? Because they are easily available, naturally existing, high specificity in their activity, and also they multiply rapidly in the presence of their host. Phage therapy is a therapeutic use of lytic bacteriophages for curing bacterial infections. Despite it's still in a minimal use, there are renewed interest in the Western side because of the developing antibiotic-resistant infections. One of the biggest complications of this phage therapy is the storage of phages for long term. Next, let's look at the bacteria that will be discussed in this research article, which is Escherichia coli, commonly known as E. coli, and also a part of our normal microbiota. However, these bacteria are opportunistic bacteria that causes infection such as urinary tract infections and diarrhea. These days, E. coli is not effective towards the antibiotics because it has turned into antibiotic-resistant bacteria. Thus, alternative therapy is highly in demand for this antibiotic-resistant E. coli. The rationale of this research article is the isolated Escherichia phage myPSH1131 could be effective against pathogenic E. coli in both in vitro and in vivo and can be stored for a long term using lyophilization method. There are three objectives in this article. Firstly, to characterize the isolated bacteriophage against E. coli myPSH1131. Secondly, to evaluate the stability of freeze-dried phages for a long-term storage. And lastly, to test efficacy of bacteriophage using G. malonella model. There are 5 essential results and discussion that were discussed in this research article. Moving on to the first one, characterization of Escherichia phage myPSH1131. The name of this phage were named using the guidelines provided by Adriansons and Brister 2015, and the phage morphology was determined by the transmission electron microscope TEM analysis. It is found out that this phage belongs to the Podoviridae family, where it consists of icosahedral head with a size 45 plus minus 5.0 nanometers and has a long non-contractile tail with a size 95 plus minus 5.0 nanometers. It's also said that this phage shares genomic similarities with other Escherichia phage, namely Escherichia phage SU10, PHI ECO 32, and also NJ01. In the host range activity step, spot test experiment was performed where 53 carbapenem and antibiotic resistant E. coli were tested, and the phage showed active lytic activity against 31 isolates from them, comprising of 9 enteropathogenic E. coli, 6 enterohemorrhagic E. coli, 3 enterotoxigenic E. coli, 3 enteroaggregative E. coli, and 9 uropathogenic E. coli. For further evaluation, double agar overlay was conducted where the phage exhibited plaques only against 29 E. coli isolates. The efficiency of plating EOP analysis data shows high productive infectivity only towards 14 isolates and low productive infectivity only against 15 isolates. One-step growth experiments were conducted, and as you can see in figure label C, it shows the phage adsorption velocity with a value of 1.6 10 to the power of -9 phage per cell per mil per min. And in the figure labeled D, it shows a latency time of the phage, which means the multiplication of the phage in the infected cell is 20 minutes, and the burst size, the release of the phages from infected cell is 130 plus minus 10 phage particles per infected cell. The stability studies of the phage shows that the phage can tolerate in both basic and acidic conditions because they have an optimum pH ranging from pH 3 to pH 13. General features of phage genome was too studied, and it was said that this phage genome consists of 76,163 base pair, where 42.3% of this phage genome is GC pair. This phage genome consists of 97 coding regions, where 31 of them are known putative functions and 66 of them are hypothetical proteins. It is said that 12 genes of this phage genome are involved in the biological processes, 15 genes are involved in the molecular function, and 4 genes are involved in synthesizing cellular components. The gene annotation also says there were no toxic-related genes in this phage genome. Lyophilization was done for a long storage of these phages, where stabilizers are used in this lyophilization and rehydrating agents were used for the recovery of the phages. Generally, sugars are known to be the best stabilizers because they are good cryoprotectants to form glass matrix to protect the phage from any aggregation. Comparatively to the other 14 stabilizers in this research use, only 3 were 2% gelatin, 1 m sucrose, and 0.5 molar sucrose plus 1% gelatin showed a stable activity up to 20 months, which concludes that they are the only strong phage stabilizing properties that they possess. And PG6000 mannitol and sorbitol shows a poor phage stabilizing property. There was only one rehydrating agent that was used in this research, which is the SM buffer, and it was known as a good rehydrating agent on the earlier studies. In vivo efficiency of single and multiple phage dose in G. melonellums. Figure labeled A shows 100% survivability when the J. melonella were injected with only saline and phage lysate with a volume of 20 microliter. And the same amount of G. melonella organisms were injected with the bacteria E. coli, and it only showed 48 hours of survivability. Thus, these two observations are being recorded as a control. Figure labeled B, where the same amount of G. melonella organisms were injected with the E. coli and then treated with only a single dose of the phage, and each showed 24 hours of survivability and gradually decreasing from the 36th hour and completely zero at the 96th hour. For multiple dosage evaluation, a maximum of 4 dosages were treated to the J. melonella organisms, and it was studied that the first 2 dosages only gave a 24-hour survivability and eventually decreases from the 36th hour as well and completely zero at the 96th hour. However, only after the 3rd and the 4th dosage, it showed 100% larva survivability even after the 96th hour. Figure labeled D shows the proof of the bacterial enumeration after each dosage, which concludes that only after the 3rd and the 4th dosage, the larva is able to survive the bacteria infected. And the summary. We got to know that this isolated Escherichia phage myPSH1131 was found to be effectively eliminating or reducing the bacteria both in vivo and in vitro. This phage has a broad infectivity against the 5 different pathotypes of E. coli that I mentioned earlier. And sucrose and the combination of sucrose and gelatin are the best stabilizers to restore the viability of the phage during the long-term preservations. And lastly, in vivo studies against G. melonella shows a very promising results that this theory can be tested in the clinical trials of the human body in the future. So that's all from me, and thank you.